Could somebody confirm you guys can hear me?
I can hear you.
Thank you. That was a bit loud. Let me turn my volume down. Okay, today is May 15th. Are there any questions before we begin? Let me share my screen here. All right, you should be seeing my screen. We are supposed to be beginning chapter 8 today. We're a little behind schedule. I don't remember how far we have to do in my chapter 6, but we're still on chapter 6. There is no lab today, so I will be logging in for 15 minutes, but to answer questions. But after 15 minutes, I'll log out. You do not need to show up unless you have a question or you want to listen to a question. Um, so, like I said, there is no lab today. Let me remind you, you should be working on your unknown work. I sent you the results to the unknown project. Well, actually, it was early Monday morning. It wasn't Tuesday night, not Monday, Wednesday morning, but I was trying to get it out a little earlier. I didn't succeed. Anyways, you got the results and should be working on your unknown project. You also need to turn in your infectious disease paper by this Saturday. Any questions about anything? Let's see, here we are on chapter 6.
Would you prefer that we ask you questions about the infectious disease during the lab portion, the 15 minutes? No, I'm sorry, not the infectious disease, the
Unknown project?
unknown project. Yes.
Probably because that project is a lab project, but if it's a quick question, I can answer it now.
I emailed you about it and I didn't quite understand. I'm not sure how many tests we should run before to eliminate, because I know we could do like a confirmation one, but how many is too many? I don't want to miss the mark by doing too little or too many.
Okay, well, as long as you don't have your unknown identified, you can continue to run tests. Too many would be you have one more test than a confirmation test, meaning once you've identified your unknown project, you can run one confirmation test. And that's assume you're doing everything correctly. You could ask for a test that doesn't help you at all. That would be an invalid test, so that would count against you. But there's no way to know that until, well, until you ask for the test, I guess.
Okay.
Okay. All right. We had talked about complex media last time. We've got a lot to do on chapter 6, so we're going to be behind schedule. Let me talk a little bit about anaerobic media. This is important for obligate anaerobes because they must avoid the exposure to oxygen. You can use a reducing media. This is a media that contains chemicals like sodium thioglycolate that actually combine with and remove oxygen from the media. We talked about that in the thioglycolate media, that in the bottom of the tube of thioglycolate, there will be no oxygen in the tube. Of course, oxygen does come in from the tube from the air because you don't seal the tube shut. And so at the very top of the tube, it'll be at full oxygen. And then it causes a gradient of oxygen going down the tube. And as you go further and further down the tube, you eventually reach 0% oxygen. The thioglycolate tube, you heat them with in the autoclave, and that will drive off the oxygen. But once you bring the tube out of the autoclave and it starts cooling, oxygen will come in to the tube, at least at the top of the tube. We have selective media. These media have chemicals in it that halt or inhibit the growth of a particular group of microorganisms while not affecting the growth of other microorganisms. An example of a selective media is 7% sodium chloride. Only organisms that can grow in 7% sodium chloride can survive and grow in this media. Most microbes cannot grow above 2% sodium chloride. But there are halophiles, organisms that like salt, for example, that can grow in 7% sodium chloride. And of course, the highest you could get would be 30%. Actually, it'd be a little bit less than that because you've got other microbes in the, not microbes, molecules in the media. But let's say 29% sodium chloride. You could probably get a media up to that. And what's my point here? My point is that only microbes that can grow in that sodium chloride can grow, and other microbes would be selected against. So 7% sodium chloride is a selective media. Any questions about any of that? All right, we have differential media. This provides differences between two types of cells. The differences are not related to how well the organisms grow on the media, and the difference is because of some additive, like a chemical added to the media, that makes visible differences between different types of cells. One example is blood agar, and on blood agar, it's differential because you can see differences in the hemolytic activity of the microbe. You can have gamma hemolysis or no hemolysis, where the red blood cells are not lysed all the way up until you have the microbes. You can't see underneath the microbes, so we don't know what's happening there, but there's no hemolysis even near the microbes. And then we have alpha hemolysis, where there's a darkening ring, very slight. Let me blow this up a little. You can see it's red up until right there where it turns dark around the off-white or white bacteria. It's also dark in the center, and that's from partial hemolysis of the blood or alpha hemolysis. And then the beta hemolysis, here we have the bacteria, they're white. And then you have a clear region. That clear region is from the lysis of all the red blood cells. So here's the blood, making it red. This is clear. You can actually see through this area of the media. And it's normal to have a alpha ring around beta hemolysis. And that's why there's a dark ring around the beta hemolysis. Beta hemolysis is complete hemolysis of the red blood cells. Anyways, blood agar is a differential media allowing you to, oops, too far, differentiate between the hemolytic activity. We also have selective and differential media. These are media that are both selective and differential. Like eosine methylene blue is both selective. It selects against gram-positive cells. So if the bacteria can grow on EMB, it's likely to be gram-negative. And it's differential on the basis of lactose fermentation. The cells, if they can ferment the sugar lactose, they will appear darkish or blackish, like that colony there, or metallic green from the fermentation of lactose. If they do not ferment the sugar lactose and they grow on EMB plates, then they will be their normal color, like this one here is either white or off-white. probably white. And that's because it does not ferment the sugar lactose. And if it's a gram-positive bacteria, it probably won't grow on the EMB plate. Any question about any of that? If it doesn't grow on the EMB plate, you cannot distinguish its lactose fermentation, so you don't know if it ferments lactose or not. We also have enrichment media. This encourages the growth of a certain group of organisms. Therefore, it is a selective media, but we use it for a different purpose, and that is we use an enrichment media to enrich for the bacteria of interest. It increases the numbers of desired microbes to detectable levels. It's particularly useful if the targeted microorganism is only a small proportion of the total population. We use an enrichment media often when we are growing microbes that we want from the soil or other environmental conditions, samples, or fecal samples. Like if you're looking for something that caused the patient's diarrhea and you want to separate it from all the other patient's microbes in the gut, you might put it on an enrichment media. And that way you can bring the sample, which is causing the patient's disease, up to high enough levels that you can detect it. For example, let's say we're selecting for, or we want a few phenol degrading bacteria from the soil, and there's thousands of other bacteria in the soil. If you were to inoculate the sample from the soil, you would have to sample over 1,000 colonies, maybe over 10,000 colonies, to just get one colony that would be phenol degrading. And that wouldn't be advantageous. It wouldn't be easy. You'd have to streak out many, many colonies before you would get the colony of interest. So you can put it in an enrichment media, such as a media where phenol is the only additive that supplies the organisms with carbon and an energy source. Therefore, only phenol degrading organisms could grow in this enrichment media. Let me see if I've got another. Now, let me go ahead and do this with paint. So let's say the organism we want, we got the wrong, yes, that's right, is something like 1 in 10,000 cells in the soil. You'd have to do 10,000 colonies just to get one of interest, and that's assuming you're lucky. If you were to put this into an enrichment media, like a media where the only carbon source is phenol, and let's say it enriches 100-fold, then we've now got it to reach to 1/100, which would be doable, not real easy. Come on. Because our enrichment was 1 in, we've enriched 100-fold. But what we can do is take a sample from this enriched culture and the enriched media and put it in another tube of the enrichment media. And now we've got it, let's say we've increased the fold by tenfold. and that's because if we did by 100-fold everything living, which is, I guess, okay, would be the phenol degrading bacteria. So I want to say just enrich tenfold. And now we only need to streak out 10 colonies because 1 out of 10 will be the cells of interest. And that would be very easy to do. You just streak out a plate, you'd probably get 10 colonies on that plate. So this would be 100-fold. If I can write that, probably not. Not real well. 100-fold increase. And then this one would be a 10-fold increase. All right, any question about enrichment media and how we use it? All right, this slide shows you the different types of culture media, just a review of what I've gone over. Let's talk a little bit about pure cultures. This is a culture that contains only one species. Um, Louis Pasteur developed a way of getting a pure culture. He did it by using liquid broth, and what he did was he made serial dilutions, meaning making very many different dilutions, and then taking 10 replicas of each dilution. And if you had, well, let's go ahead and open up this one. And if you had one dilution where one tube out of 10 tubes had growth in it, Louis Pasteur correctly assumed that this culture was likely to be a pure culture. This is a very difficult way of getting a pure culture, but it was the first way microbiologists had. Um, then Edward Koch came around and he developed an easier way of getting a pure culture, and that is you have a solid media, and then he streaked out his bacteria on the solid media, and eventually he was streaking out single cells, which would grow up to be a colony, an isolated colony. And then he would take cells from that isolated colony and streak it out a second time on another plate, getting two colony isolation events. So he passed the cells through two colony isolation events, and he correctly assumed that the chance of it being a pure colony was quite high. It's actually greater than 95%. And so if you streak it out two times through two colony isolation events, the cells are likely to be pure. It doesn't guarantee they'll be pure, but there's an over 95% chance that they will be pure. And that's such a good way of getting pure cultures. We still use that way in microbiology today. Any question about any of that? A colony can arise from a single cell, but they can arise from two cells or a group of attached cells, meaning a clump of cells. That means when you streak out the plate, not all of the colonies will be pure because some of them will be formed by two cells or a clump of cells. And so when we streak out the plate of colonies, we call them colony forming units that form the colony. And then we don't count the number of cells by counting the number of colonies. We count the number of colony forming units. Any question about any of that? Because we don't know the exact number of cells because some of the colonies are formed by more than one cell. To obtain single colonies, which is a critical part of attaining a pure culture, you streak for colony isolation and you pass the cells through two isolated colonies, meaning you streak out your mixed culture. I guess that's the one there. And then you sterilize the loop, streak it out a second time, sterilize the loop, streak it out a third time. And someplace along the line, we're hoping, let me see if I can blow this up, that you'll get isolated colonies. As you can see here, this culture we initially streaked out contained at least two different cells because we have two colony types. And these are easy to determine because they have different um, colony characteristics. They have different colors, for example. Now, if they had the same color, it'd be much more difficult to obtain isolated colonies because you wouldn't know, not difficult to obtain isolated colonies, difficult to know if you had more than one culture. We'll word it that way. What you do is you take from a single isolated colony and then streak it out on another plate, getting isolated colonies. And you take from a single isolated plate colony on the second plate, and then you set up your stock culture from that isolated colony where the cells have gone through two colony isolation events. And then your chance of that being a pure culture is over 95%. You never get 100% just because of the way, I don't know, statistics run or biology is. And I once had a friend who was isolating out a nitrogen fixing bacteria or something like that, a nitrogen metabolizing bacteria. And he didn't know it, but he had to have two species of bacteria present because he wasn't supplying the correct form of nitrogen to get the bacteria of interest for him. And so every time he streaked it out, even though he thought he had a single colony and he sent it through two colony isolation events, he had to have two different species there to get the bacteria to grow on the plate. So he never got an isolated colony with one species on it. Um, I think what he did was he finally, when he must have sequenced it because he knew that he had two species there. And what he did was he streaked it out on an enriched media, something like blood agar, and he finally got two species to grow out of a single colony that when he was growing it on a media where he wasn't supplying nitrogen. My point is, you never can be certain you have 100% chance you have a pure culture, but if you do it through two colony isolation events, your chance of it being a pure culture is greater than 95%, which is good enough for microbiology. Any question about any of that? All right. Remember, microbial growth refers to the increase in numbers, not the increase in cell size. There are three ways that microbes can grow, including by bacteria. Most bacteria grow by binary fission, where the cell literally divides into two. So one cell, it then splits down the middle, dividing into two. There are some bacteria, not too many, but there are some that grow by budding, where the parent cell will put out a little bud, and then that bud will grow bigger and bigger, and that bud will not become isolated from the parent cell until it's grown to about the size of the parent cell. I've got a picture of that. I'll show you that in a minute. And then there are a few bacteria that reproduce by reproductive spores, usually at the tip of a filament bacteria, meaning a bacteria that looks like a filament. And I'll show you what I mean by that. Oops, wrong one. So the bacteria aren't really rods. They're more like filaments. And I can't really draw a filament, so let's just make it something like that. And what happens is this filament will stand up. Let's make it something like that. It's a filament, not a rod. And then it'll put out reproductive spores. The reproductive spores are almost always at the tip of the filament, standing up, so that when wind or some animal comes by, the reproductive spore can be separated from the parent cell and then germinate in another location to give rise to another parent cell. And that is a reproductive spore. The reason why it is reproduction is we had one cell, and now when we have the reproductive spores, we have got 5 cells because there's 4 reproductive spores. Any question about that? So reproductive spores is reproduction. Remember an endospore, where one parent cell forms one endospore inside of it when the environment's decaying, and then the parent cell dies as the environment decays. So I'm breaking the cell wall because the cell is dead. We'll break it here too. And then the reproductive spore comes out of the parent cell. So a reproductive spore, that's not a very good arrow. It's a little better. A reproductive spore, we had one parent cell, which will die, and then there's one endospore. That's not reproduction. One cell equals one cell. Remember, an endospore is a survival strategy. The endospore can survive harsh environmental conditions, waiting for the conditions to become favorable, in which case the endospore will germinate and form a parent cell, and the parent cell is metabolically active and can reproduce, forming another parent cell, meaning a vegetative cell, not an endospore. Any questions about the difference between a reproductive spore and an endospore? All right, so let's take a look at binary fission. What happens in binary fission is first the DNA replicates and then separates, where one DNA molecule goes to one end of the cell, the other DNA molecule goes to the other end of the cell, and then the cell literally starts dividing in two, where the cell membrane and the cell wall starts growing in the middle of the cell. And then the cell will eventually separate as the cell membrane and the cell wall divide the cell. And then the cells will go on their way. They will elongate a little bit. And then they will reproduce by binary fission. Here you're looking at a electron microscopic image of a rod, which is actually in the process of going through binary fission. There's the partially formed cell wall and cell membrane. Any question about any of that? All right, we do have a few bacteria that bud. Here's a little bud that's being put out on the cell. There's a little bigger bud, and there's an even bigger bud. The bud will grow until it becomes close to the parent size, and then the bud will, what do you call it? Break off from the parent cell. I got a little movie on that. So the bud will form right there. There's the bud beginning to form. The DNA is actually reproducing right now. And there we can see the DNA moving into the bud. The bud gets bigger, and about here, I got that too far. About there, the bud is beginning to separate meaning it's starting to close off. It's not totally closed off. About right there is where the bud is now sealed off the bud from the parent cell, and you can see the cell wall and the cell membrane um, of this cell as well as that cell. And then as we go further, this cell will put out a new bud right here, and I think this cell will put out a bud right there. There you can see the bud beginning to form there, and there it's beginning to form there. Any questions about budding? Only a few bacteria reproduce by budding or by reproductive spores. Most of them reproduce or divide by binary fission. When we're talking about the growth of bacteria, it's important to understand the generation time. The generation time is the time required for one cell to divide into two. That is also the same time for the population numbers to double, meaning if you have a culture with 1,000 cells in it, its doubling time would be the time it takes for the culture to go to 2,000 cells. And that's the same as for one cell in that culture to go to two cells. The generation time varies considerably between different species. They can be as fast as 20 minutes. Most bacteria, the doubling time is 1 to 3 hours. For Mycobacterium leprae, the doubling time is very slow, and it takes 10 days for one cell to divide into two, or for the population numbers to double. The reason is because Mycobacterium have a lot of mycolic acid in their cell wall, and it takes the cell time to make the mycolic acid. Note when we discuss the generation time as 20 minutes, it's implied 20 minutes per generation. So most bacteria's doubling time is between 1 and 3 hours per generation. That per generation is understood. When we're talking about the generation time, don't get confused on what the number mean. It is time per generation. It's not generations per time. When we talk about like miles per hour, we're always saying how many miles you traveled per unit of time. For the generation time, it's not per unit of time, it's time per generation. And that gets students confused, probably because they're just so used to talking about things in miles per hour. Any question about any of that? E. coli is one of the fastest microbes we have, and the generation time of E. coli is about 20 minutes per generation. Remember that per generation is always implied. When we're measuring the generation time of bacteria, microbiologists like to use a semi-log plot, where we don't plot the arithmetic number on the y-axis, because if we did, this one is the, oh, excuse me. This one is showing you the mathematical arithmetic number on the y-axis. And then the generations is the mathematical arithmetic number here. But that's why it's semi-log. If we were to plot the number, the growth of the cells using the regular arithmetic model, we'd see something like this, a curve like that, which is a very difficult curve to explain. It would be something like, let's see if I can remember, y equals mx squared plus nx plus B. It might be even B plus Y, something like that. Not Y, but W, something like that. It's a complex mathematical formula to explain this curve. And then all the points are right down here. We have one cell dividing into two, two into four, four into eight, eight into 16, 16 into 32. So most of the points, plots on this graph, are all together right here. And then the next point, after, I don't know, 100,000 cells, it's not going to be on the graph. It's not going to be on the piece of paper. It's going to be someplace on my ceiling. And that's because it is a um, showing exponential growth. Any question about any of that? All right. If we use a semi-log plot where the generations is actually a regular arithmetic number, but we plot the number of cells on a log plot, then we get a curve like this, which is a much simpler mathematical equation. It's simply y equals mx plus b. And then the points will be spread out better. You don't have a whole bunch right here. I don't know what it would be because this is a log plot, but you'd have, let me see, log of base 10, 1. Well, I guess that's 10, I think. So you'd have something like 1 into 2, those points are together, 2 into 4, about halfway, and then 4 into 8, almost up to 1. And then you get 16, that's close to 2. 32, that's about there, something like that. My point is you don't get all the points here. And then to get the next point, you can just extend the graph. The next point's going to be about right here. It's not going to be on the ceiling. It's going to be actually on the sheet of paper, the end of the piece of paper, but about right there. Any questions about any of that? So those are two reasons why biologists like to use a semi-log plot, and well, actually three reasons. That is, you can get the points here spread out, and all the points are all together here. And then the next point can actually be on the graph piece of paper. And then the equation is actually much simpler than this equation. Any questions about any of that? Why is that there? When we're talking about the phases of growth, bacteria almost always have this growth curve. And in fact, whenever you look at organisms which are introduced into a new environment, they also tend to follow this growth curve. And we know this because somebody introduced a bunch of deer on an island. I think it was on the East Coast. I don't remember what state it was off of, but they introduced deer onto the island. And initially, the deer were in a lag phase. They were new to the island, they didn't know the island, and so they weren't interested in reproducing. They wanted to explore the island. So there was an initial lag phase, as well as there was, I suppose, the concern of the deer that the person came and captured them, probably put them to sleep and captured them, and then moved them to the strange island, move them out of their normal environment that they knew. And so the deer explored the island, entered the lag phase, and then they found lots of food on the island, and they had an exponential growth phase where the deer started reproducing. Initially, there was lots of food around, lots of space, and so the deer population took off exponentially. Eventually, the population started slowing. And that's because there was less food around, lots of deer around, less space around. The population number then became stationary on the island. And that's because some of the lucky deer found food and could reproduce. The unlucky deer didn't find enough food and started starving and started dying. And so the number of deer reproducing was equal to the number of deer dying. And then as the food on the island decreased even further, the population entered a death or decline phase where near the end, all of the deer were not reproducing. They were all starving and starting to die. Eventually, if they followed that further, and assuming that the growth on the island could not grow quick enough to keep the few deer alive, depending on what time of the year it was, I guess, all of the deer would eventually die. This happens in bacteria too. When you take the bacteria from a solid culture and put them in a liquid culture, initially the bacteria enter a lag phase. In the lag phase, they're making the enzymes that they need to grow in the new environment, which is a liquid culture instead of the solid culture. And then the bacteria have the enzymes they need, so they enter the exponential growth phase, where initially there's lots of room for the bacteria to grow, there's lots of nutrients around, and there's very few, if any, waste products around. But as the population grows, there will be less space, there will be less nutrients, and there will be more and more waste products. And so the population growth will start to slow. Initially, Eventually, the population numbers in the population will be stationary because the lucky bacteria will have some nutrients around, not too many waste products around, but there will be some, and they'll have a little bit of space. And the lucky bacteria can grow and reproduce. The unlucky bacteria will have very little nutrients around, if any, lots of waste products around, and very little, if any, space. And so the unlucky bacteria will die, and the growth will equal the death. So the birth and equals the death, and the population will be stationary, won't change. As the population ages, there will be more waste products around, fewer space around, fewer nutrients around, and the population will begin to decline, where the deaths are higher in number than the births, at least initially. There are very few bacteria that'll be very lucky and they have enough nutrients around and not enough waste products around, a little bit of space that they can actually grow. So there will be initially some growth, but there will be more cells dying. So the population number declines. Late in this decline phase, there will be no cells growing, and there will only be cells dying. How long the cells take to die will be dependent on how sensitive the cells are to the decaying environment. Cells like E. coli will not live longer than about 2 weeks, and that includes in the stationary phase, maybe even part of the log phase. I don't know. After about 2 weeks, all the cells in the media, liquid broth, will die if it's E. coli. However, if there's cells that form endospores, these cells can survive, and they can survive for at least 8,000 years because we have endospores which have germinated from the mummies they've obtained from ancient Egypt. And I think the longest was 40,000 years, which was an endospore they got from a Antarctic ice drill. They drilled into the ice, went down many feet into the ice, and they found an endospore frozen in the ice from 40,000 years ago that they were able to thaw out and actually get to germinate. And that's the longest we've had for an endospore. Any question about any of that? All right. So know the 4 phases of growth: the lag phase, the log phase, the stationary phase, and the death phase, and what causes each of the different phases of growth. Any question about any of that? All right, we have different methods for measuring microbial growth. there are direct methods and indirect methods. The direct methods actually have a direct method for obtaining a count, like you could do a plate count or actually count the number of colonies that form on the plate to get an estimate for the cell numbers. You can do the filtration method, which is somewhat similar to the plate count. You just pass the cells through a filter, put the filter on a plate of media, then grow up the filter or the colonies on the filter. You have the most probable number, and you have the direct microscopic count. We're actually doing a direct count of the cells under the microscope. We have indirect methods where we can get an indirect estimate of the number of cells in our sample. There's the turbidity meter, which measures the amount of light coming through the media. There's metabolic activity that measures the metabolic activity of the cells. And then there's dry weight. In both metabolic activity and dry weight, you get higher numbers if you have more cells. In the direct method, the plate count is the most common method used in microbiology, which measures the number of viable cells. This method and the filtration method are the only methods we have for distinguishing viable cells from non-viable cells. Anyone have a question about that? All right, so in the viable plate count, you plate the cells on a plate, and only the viable cells will grow. The non-viable cells will not grow and form a colony. And so that's how the viable plate count only counts viable cells. It takes about 24 hours or more for the colonies to form, and the results are reported not as cells per mil of your original sample, but colony forming units per mil, because when we count the colonies, we don't know if the colony is formed by one cell, two cells, or by a clump of cells. And the plate count is frequently called the viable plate count because it only counts viable cells. Any question about any of that? All right, so how you do a viable plate count is you make serial dilutions from the original sample. You might sample the original sample too, especially if you think it might be few cells per mil, but we can take the original sample and put 1 mil of it into 9 mils of media or water, whatever you're diluting it in, and then mix those cells up. Take 1 mil out of that, put it into 9 mils, mix those cells up, take 1 mil out, put it into 1 mil, etc. And then we have 1 to 10 dilution, 1 to 100 dilution, 1 to 1,000 dilution, 1 to 10,000 dilution, 1 to 100,000 dilution. How you got 1 to 100 was we have 10 dilution here, and then you take 1 mil, put it into 10, that's 1/10 of this dilution here, and that would be 1/10 times 1/10 or 1/100th. Any question about that? You then plate an aliquot from each of these dilutions on a different plate, and it depends which method we're using, the pour plate method or the spread plate method, for how much you put on the plate. In the pour plate method, you normally put on 1 mil, and then you pour that 1 mil, spread it around the plate by swirling the plate. More likely, what you do is you just tilt the plate back and forth so that 1 mil moves everywhere, and then you go this way and that way. And you could go that way and that way too. I don't know if you saw that, but I was trying to show you how you tilt the plate back and forth. This, they're swirling it to get the 1 mil to go on the plate. You'd probably need a special device for doing the swirling. The point is that this then is spread on the plate, and then you incubate it after it's absorbed, and then put it in the incubator. And then after a period of time, like 24 hours, take it out of the incubator. And then you have colonies on the plate. Hopefully they're isolated colonies. And then you count them. The spread plate method is you usually use much smaller inoculant, like 1/10 of a mil, and then take a spreader using a clean spreader, I mean you've sterilized it or um, disinfected it some way, and then spread that 1/10 of the mil over the plate, and then you incubate that, pull it out, and then count the colonies there. So they're very similar methods. The difference is really just how much you're putting on the plate and then how you're spreading the bacteria. So from each dilution, the original would be even more concentrated here. You then count the colonies. This dilution is too many to count. This dilution, too many to count. This one, you could count some of the colonies, but you couldn't count all the colonies. So once again, too numerous colonies to count. This one, you get, I don't know, 30 to 40 colonies per 1 mil that we put on the spread plate. For this one, it would be very easy to count. You're only getting 4 colonies on the plate. Which of these do you use? Let's say you got 40, 42, and this one you get 4. Well, you want to use a plate where you have something like 30 to 250 colonies per plate, as opposed to one with just a few colonies per plate. The reason being, in the show here, open up a new. The reason being is if you had, let's say, 200 colonies, colony forming units per mil, let's go colony forming units, in that dilution, and let's say it's dilution 1 to 10,000. Well, let's go 1 to 1,000. That way it'll be easy. And then 200 times the dilution, which is times 1,000, equals 2 times 10 to the 5th. Not seeing how to do that here. Let's see if this does it. No, that won't do it. Oh, there it is. Make this a little bigger so you can see that. So the original sample will be 2 times 10 to the 5th colony forming units per mil. Any question about that? All right. Well, if we had the thing like 4 colony forming units, oops. And this would be the dilution 1 to 10,000. So that would be 4 times 10,000. Oops, don't have that right. Gotta put an X in there. 4 times 10,000, which will be is that right? Yeah, we'll go. 10 to the fourth. Usually these numbers would be a little bit closer together than that. So maybe what I should do is go 4 over 100,000, and it'll be a little closer. Now make it 10 to the fifth. Oop. And which number do you use? Well, you use this one because the error here will be plus or minus. Let's just go plus or minus 1. If we go plus or minus 1 here, oops, I got that underlined right there. That's plus or minus 25% here. And that's because 1 out of 4 will be 25%. Let me get that plus or minus. And this plus or minus here is going to be plus or minus 1 out of 200, which will be 0.5%. I don't have the percent sign in there. Come on, number. We want a mean which is small and has very little variance, and this will be a much smaller variance, 0.05%, than the 25% mean. Any question about any of that? So the point is you use colonies between 300 or 30 to about 250. You want to use a dilution, where the number of cells is between 30 and 250 to get this mean having a small error. If you use a small number, the plus or minus 1 will be a plus or minus a very large number, 25%. I don't have that minus in there. I mean underline there. There we go. Well, kind of. I guess I did have it. Just couldn't see it because the blue line. Any question about any of that? All right. The filtration method is similar to the viable plate count. The difference with the filtration method is it can get you a count of the colony forming units when the bacteria are very dilute in number. Like if you're sampling bacteria in public drinking water, you wouldn't want to do a plate count because if you did a dilution, you're going to get nothing because the bacteria are so low to begin with. And even if you were to do the original bacteria, meaning 1 ml of water, will be likely to have very few bacteria in it. I don't know what it is, but it's very low. It'd probably be something like 4 to 10 if you do the original ml of water, put it on the plate. It might even be less than 4, something like that. I don't know what it is, but it's very low. And so what you'd want to do would be to send the water through a filter and send in a large amount through, maybe a gallon of water, maybe a cup of water, I don't know, something like that. Send it through the filter where the bacteria will get stuck in the filter. The water will go through the pores in the filter. And then you put the filter on the media to let it grow, and then you count up the colony forming units on the filter, which you can see here. Here we're seeing the filter, and it looks like in this case, there's very few colonies that are formed by one cell. They're mostly formed by clumps of cells, a few by pairs, diploids, diplets, whatever. Another direct method is the microbial count, where you actually count a volume of liquid under the microscope. Cells are put under the microscope and then counted. It's not good for mobile bacteria because if the bacteria move and you're counting, let's say this square here, well, this one here, once you count it, can move out of the square. And then let's say this one here moves into the square before you count it. So it's not good for counting motile cells unless you kill the cells and then count them. And it cannot distinguish between a viable cell and a dead cell. Like when we look at this cell here or there, can you tell if it's viable or not? You just can't tell if it's a viable cell or a dead cell. Microscopic counts always use a special slide, which is gridded, and it allows a certain volume of liquid underneath the slide. Specifically, usually these slides allow 1 microliter for one of these bigger squares right there. And so if you counted these cells here, then you just multiply these cells times 10 to the 6 because this is 1 microliter or 1/10, one, whatever. Like I said, normally that doesn't seem to make sense because it's per mil. So maybe it's less than a microliter, but you normally multiply this by 10 to the 6, the count in this square here, and that's because the volume here is a certain volume, something like 1 microliter. And there's a special gridded slide which allows a certain volume underneath the slide. And then it has grids which also allows only a certain volume under the grid. Any question about the direct microscopic count? The only thing hard about the microscopic count is it is a bit of an eye strain for the person who has to do the counting. But it's fairly quick. You just take your sample, put it under the microscope. You could take a dilution of the sample if your cells are too concentrated. Put it under the sample and then do your count. I'm not going to talk about the most probable number. It also is a direct method of counting cells. I've actually never used the direct most probable number. I have done plate counts, I've done filtration, I've done direct microscopic counts. I've also done the indirect methods, turbidity, metabolic activity, and dry weight, but I've never done most probable number, so I'm not going to talk about it. You can read about it in the book. In the indirect methods, turbidity measures the amount of light going through the media. And you first measure the media without any cells through it, and there will be a certain amount of light coming through the media. You measure that amount of light coming through, and then you measure the amount of light of the media containing the cells. The more cells there are, the more light will get blocked. And then you measure the amount of light getting through, look at a chart, and it'll tell you how many cells per mil you have in the media. The only hard part about this indirect media, the turbidity method, is to make the chart, you have to make a whole bunch of different dilutions of different cell numbers. You have to know the number of cells in each different dilution. And then you measure the amount of light coming through. And then you make this chart. And if there's no cells, this amount of light coming through means there's 0 cells. And if there's a little bit of cells, then you get less light coming through. And then you have lots of cells, only a little bit of light will get through. And then you know the number of cells here. And so you look on the chart and you make that measurement. And the thing is, once you get the chart set up, you can then use it indefinitely for the rest of the chart's life. You just measure the amount of light coming through the media, compare it to the amount of light coming through the media with cells, and then you look on the chart and it tells you how many cells per mil you have in the media. Boom. Any questions about that? And how the light is measured is you use a spectrophotometer to measure the amount of light. The more cells you have, the less light will come through the media. A metabolic activity assumes that the bacteria numbers make a metabolic product like acid or CO2, which accumulates in direct proportion to the number of bacteria. So the more cells you have, the more CO2 you have. And if you took Biology 160, you may have done that experiment where you actually measured the amount of CO2 coming out of the bacteria. I've done this where I measure the amount of CO2 coming out of the bacteria. And initially, you have to count out how many cells you have, measure their CO2 coming out over a certain amount of time, like 10 minutes or an hour, depending on how your chart's set up. And then you make your chart. The good news is once you make the chart, you can then use this chart indefinitely for the life of the chart. And what you do is you just have a number of cells, you don't know how many, and then you measure the amount of CO2 that's produced, compare it to the chart, and it tells you how many cells you have that made that CO2. Any question about that? So the more cells you have, the more CO2 you get that accumulates. I've also done the dry weight, where the method is to grow up your cells, usually in a liquid culture, centrifuge them down, pour off the supernatant, grab your cells. Usually you allow them to dry by putting them in a, oh, what do you call that? A suction aspirator, pulling out the air and pulling air through so it dries the cells. You see, like have a filter here and then you pull the water this way, the air this way, and then the cells are on this side of the filter, and then you dry it. And once it's dried, you can then weigh the cells. The more cells you have, the more weight you will have. Initially, to make the chart, you have to have a known concentration of different cells. So like you have no cells, you have no weight. You have a few cells, you have very little weight, like, I don't know, maybe 1 microgram. And then you have a certain number of cells, like 10,000 cells. You know, it's, I don't know, 1 microgram. I don't know what 10,000 cells weighs. But you have to know your number of cells and then do the weight to make the chart. And then the advantage is once you have the chart set up, you can just weigh your cells, look on the chart, and it tells you how many cells you have. And like I said, you have to do this drying the cells. The advantage is that when you grow up your cells, you just dry them and then you can weigh them at your leisure whenever you have time to to do it. You don't need to dry them or weigh them right away. You could probably even keep them for a year before you weigh them. You wouldn't want to do it that long, but you could if you needed to. Any question about the different ways we have for estimating bacterial numbers? All right, that's it for chapter 6. We're way behind schedule. Let's begin chapter 8. Here I have the major goals and the lesson outline. Always know the terms of the chapter. I'm not going to go through it because of the time. Be able to describe what DNA replication is, RNA transcription, and protein translation. And then 3, understand the different types of mutation. Chapter 8 is covering genetics. We'll be speaking specifically about microbial genetics. This is a chapter summary. We're first going to just talk about the structure and function of the genetic material. We'll talk about the flow of genetic information. We'll talk about DNA replication, RNA transcription, and then protein translation. We'll then switch gears and talk about the regulation of bacterial gene expression using an operon to discuss bacterial gene expression. We'll then talk about mutations, the different types. We'll talk about genetic transfer and recombination. And then lastly, for the last slide, we'll talk about genes and evolution. Any questions about anything? All right. Know what genetics is, the study of what genes are, how they carry information, and then how that information is expressed and how genes are replicated. Know that a gene is a segment of DNA that encodes a functional product. The functional coded and encoded is usually a protein, but it could be an RNA. It just depends on which gene it is. Like if it's a tRNA gene, it's only going to code for RNA. But if it's a gene that codes for protein, it'll first code for messenger RNA, and then that messenger RNA will code for protein. Any questions about that? I'm going to blow this slide up. So there we're looking at a chromosome containing the DNA, and on the DNA, we have a region which is called the gene. This is, of course, a eukaryote cell, and how I know that is because we have non-coding regions of the DNA that does not code for a gene. Any question about any of that? And there we have a chromosome showing you the different locations of certain genes on the chromosome. That's actually the human X chromosome. If you look someplace, there'd be, I don't know where it would be, but there would be, not sickle cell anemia, but where the child who's born can't clot his blood, a hemophilia. Come on. Not sure what happened here. Some more terms. Genome is the study of what genes are. I've already talked about genetics. There we go. Genome is all the genetic material in a cell. When we're talking about a microbe, it's all the genetic material in an organism as well. The study of genomes is called genomics. There are 2 terms in genetics you need to know. The genotype is the genes of an organism. For humans, we're diploid, and so the genotype for blue eyes would be genotype lowercase b, lowercase b. But the genotype for brown eyes would be uppercase b and lowercase b, or uppercase b and uppercase b. So that would be the genetics of eye color. The phenotype is the expression of the genes. So the expression of lowercase b would be blue eyes. The phenotype of uppercase B, lowercase b would be brown eyes. So you should know genotype and phenotype. For diploid organisms, we'll always have two genes for that phenotype. The exception would be if the gene is on the X chromosome or on the Y chromosome. For boys and men, you'd only have one gene. But for women, you'd actually have two genes. What's an example? For hemophilia, why hemophilia? If it's carried on the X chromosome, it's much more common in boys than in girls because you only need one gene in the boy to get hemophilia, but in the girl, you'd need two recessive alleles to give you hemophilia because the normal gene, which is not hemophilia, is dominant over the hemophilia allele. Any question about any of that? All right. Here we're looking at a chromosome, the strand of DNA, which wraps up to form the chromosome. And this actually is an unwrapped chromosome because it's a bacteria, but the point is on one region of that DNA, we could have the genes, and there's the chromosome. Remember, a chromosome includes both DNA and protein. In a eukaryote chromosome, the main protein is a histone protein, but histone proteins are lacking in bacteria, but they do have other proteins in the chromosome. All right, DNA is a polymer of nucleotides. There are 4 nucleotides in DNA: adenine or A, thymine or T, cytosine or C, or guanine or G. And you can learn them in either name, and you can either say T or thymine. DNA is a double helix, meaning there's 2 strands. There is proteins on that double helix to make the chromosome. DNA is hooked together in one strand, is held together by the covalent chemical bond holding the sugar to the phosphate. To the sugar, the phosphate is the backbone of the DNA, holding the single strand of DNA together. The nucleotide bases are not part of the backbone. The nucleotide bases bond to the other strand of DNA by hydrogen bonds, and they're sort of the steps in the DNA ladder. So your DNA is 2 strands, but each strand is only held together by hydrogen bonds. The hydrogen bonds are held together by the nucleotide bases. AT always has 2 hydrogen bonds. CG always has 3 hydrogen bonds. We'll talk a little bit more about that in our next slide or next day. But the 2 strands of DNA do not run in the same direction. They are anti-parallel to each other, meaning one end is 3 prime, the other end is 5 prime, the other end is 5 prime here, and then 3 prime here, meaning they're running in opposite directions. You don't need to know about the 3 prime. It refers to that this carbon here is the 3rd carbon binding to the phosphate. Let me think about that. I used to know this. I'm trying to remember. No, this is the 3 prime carbon here. The 3rd carbon when chemists count the carbon in the bond. And this one here is the fifth carbon. And this is why this is the 5-carbon end of the DNA, because it ends with the 5-carbon here. And this is the 3-carbon here. So it begins with the 3-carbon. And you don't need to know about the 3' and the 5'. You just need to know the DNA strands are anti-parallel. All right, any question about anything I've talked about so far? If there's no questions, I'm going to end it here. And if you have a question, I will be in the lab at 6:30 for 15 minutes. After that, I'll log out unless there's a bunch of other questions. All right, I'll see you.